Hello everyone, thanks for being here. So, great talk by Hasiban. He gave a really nice background on the infectious disease vaccines, especially mRNA vaccines and how they're designed. So earlier this year, in 2024, we got an opportunity to write a review in Nature Reviews Drug Discovery on a lot of developments have been done in the field of vaccine development, especially infectious disease in the last 4 years. And we were able to write a review summarizing all those developments and what it tells us how to design next generation of vaccines. So why is this still a hot topic? Basically, we have seen that how successful these vaccines have been in saving millions of lives. This was a report published recently by WHO where they have shown that extensive immunization efforts throughout the world since 1974 have saved lives of more than 150 million children in the past 50 years for these particular vaccine platforms. And more recently, the efforts in the field of nano and nucleic acid delivery have shown how within 1 year of discovery of the SARS-CoV-2 genome, which was released, and like these vaccines were developed, the mRNA vaccines, and how billions of doses were administered, and how they were able to potentially save the world from such a panic. But all is not done in this field of vaccines, and there have been several challenges in the field to develop next gen of vaccines. One of them has been to reduce the cost and basically reducing the cost of each dose of these vaccines so that we can not only lower the cost of vaccines but also improve the accessibility throughout the world. Another challenge is to develop these vaccines which can generate broadly neutralizing immunity, and this could be really important for vaccines, for making vaccines against pathogens such as HIV, making universal influenza vaccines so that you don't need to get like different vaccines each year, and also making pan-coronavirus vaccines. Other aspect is the reactogenicity that has been associated during the last 4 years, especially with the mRNA vaccines that have been developed. There have been reports of like how these vaccines, especially after the booster dose, there were instances of fever, chills, and like muscle pain, which prevented these a lot of the population to get like subsequent booster doses. So having a vaccine platform that minimizes these side effects can be critical to improving the overall coverage in the vaccine population, and hence promoting the ability to induce like herd immunity. Finally, other administration routes such as like nasal vaccines or other vaccines routes could be really important, such as when you have the ability to make nasal vaccines, they not only provide you immunity, but they can also prevent transmission of these respiratory viruses at a very early stage. Hence, they could be even more effective. So these are a few challenges, and when we wrote this review, we had these things in mind. So just a brief overview of like what a vaccine basically looks like. We have 3 different components in a vaccine formulation. You have one is the delivery component, which in case of mRNA vaccines was of course the lipid nanoparticles, but other vaccine platforms can utilize other delivery components that could be polymers or other nanoparticles. Secondly, for the antigen, it can be an RNA which can encode for the viral proteins or tumor neoantigens for cancer vaccines, but it could be just these recombinant proteins for the viral antigens. And finally, a lot of vaccine platforms use different adjuvants, such as the recombinant proteins, especially they use adjuvants such as matrix M or other different kinds of, such as alum and all other different kinds of adjuvant platforms. So I'll talk about, in our review, we covered all these design aspects. So I'll start with the strategies for designing the antigens. So there are a few concepts regarding like designing these antigens which we covered. One of them was like designing the antigen which has appropriate conformation. For example, like in the FDA approved vaccines that have for RSV and SARS-CoV-2, it was found out that having an antigen that was in pre-fusion conformation yielded much stronger neutralizing antibody and a protective immune response as opposed to a post-fusion antigen conformation. Secondly, in the field of antigen discovery, people have found out that, for example, this was more relevant in case of HIV vaccines. They found small neutralizing epitopes, and they made multimerized nanoparticles using these epitopes, such as an EOD-GT8 antigen. And also there have been phase 1 clinical trials where they made multimerized antigens for the hemagglutinin stem nanoparticles. And using these multimerized nanoparticles generated a much superior neutralizing antibody response. So this is another strategy for the antigen development. Finally, when you take these individual antigens and you divide them into small immunogenic peptides, you can classify them on the basis of which peptide epitopes can generate a T cell response and which peptides can generate a B cell response. And hence, you can generate like HIV T cell vaccines or influenza T cell vaccines. You can generate the kind of vaccines depending upon what kind of adaptive immune response that you want. The more successful examples of these have been seen in the T cell vaccines that have been developed by BioNTech and Moderna. Finally, there have been several different RNA-based platform, nucleic acid-based platform that can actually encode for all the previous strategies in themselves. And depending upon which platform you're using, ranging from RNA to DNA, you can have different levels of antigen expression and different levels of immune activation. And also you can engineer these RNA platforms to basically encode for example, like a multivalent antigen, which can generate a more biased B cell response, and this is a strategy that is being currently used by Moderna in their phase 1 clinical trials for making an HIV vaccine. Finally, all the transmembrane antigens, all the FDA-approved COVID-19 vaccines had these antigens that were expressed on the membrane of the cells, which kind of mimics the way a virus replicates inside a human antigen-presenting cell. So that is also another strategy that people have used. Finally, people have also used these fused antigens. This has been done more in context of cancer vaccines, but there's also a potential area which can be used to develop next generation infectious disease vaccines. Now to talk about the next generation delivery vehicles, I'll hand over to Kaelan.
Thanks, Akash. So yeah, now that we've sort of decided what's inside the vaccine, how we're going to encode the antigen of interest, the next thing that we need to consider, which is of interest to a lot of the people here in the nanomedicine community, is sort of how we're going to deliver these antigens and elicit an immune response against them. And so what that really requires is considering the delivery vehicle that we're going to use. And so here we've highlighted a number of different delivery vehicles which have gone into clinical trials. There's protein nanoparticles, which are sort of a fusion with an antigen, VLPs, polymer nanoparticles, or LNPs and liposomes. And when you're developing this delivery vehicle, there's a lot of things to consider during the development of it to make sure that it works effectively for vaccines. You need to study sort of how it delivers the antigen of interest by sort of evading degradation by the innate immune system so that it can reach the lymph node and elicit adaptive immune responses, recruiting the correct cell types. You need to consider things like antigen multivalency, which may enhance immune responses, as well as look at the different immune cells that are recruited. Is this delivery vehicle causing uptake in antigen-presenting cells or other innate immune cells of interest that are then leading to an immune response? And what are the sort of vaccine kinetics that are resulting from this delivery vehicle? Does it give long enough expression or presentation of the antigen to lead to effective immune responses? And ultimately, when you consider all these factors together, hopefully you can come up with a delivery vehicle that leads to strong humoral and cellular immunity. So looking forward, sort of what are the challenges and future directions for vaccines? And hopefully we can maybe have a discussion about this afterwards. One of the challenges that just talked about is coming up with better delivery vehicles. We have LNPs. The LNPs that were used in the COVID-19 vaccines were sort of what were available at the time. They were pushed into the clinic and they worked pretty well, but there's definitely still an opportunity, as Hasib talked about in his talk, to improve those delivery vehicles, not just LNPs, but other delivery vehicles as well. And as Akash talked about, there's more opportunity to improve antigen designs. Right now, for the most part, a lot of antigens are either just the whole virus itself, or we've chosen a subunit from that virus to act as an antigen. But there's definitely an opportunity to make de novo antigens that are either for T cell vaccines for things like cancer, or unique antigens that can generate broadly neutralizing antibodies. And there's also a need to better understand immunology. You think we know a lot about how the immune system responds to vaccines, but there's still a lot of opportunity to improve our understanding. Things like, why do certain members of the population respond really strongly to vaccines while others do not? And another thing that would be great is, can we predict how a vaccine is going to work in a 96 well plate, in a 384 well plate, instead of having to go into animal models? That would really sort of accelerate the development of vaccines. And then, going back to this idea of accessibility of vaccines, there's an opportunity to improve their availability globally, either through improved thermal stability, lower cost, and considering different administration routes in other cultures where getting an injection may not be the preferred route of vaccination. So let's say you got really inspired by this talk and you want to go start working on your own vaccine project. We wanted to highlight a few resources that are available to sort of get you started on that research. One that I wanted to highlight is this website called BEI Resources. It's run by both the NIH and ATCC, and it's a huge repository of like thousands and thousands of reagents that are provided to researchers free of cost. So they have basically any antigen that you can imagine. They have peptide pools for stimulating splenocytes, all sorts of stuff. They even have influenza virus, like if you wanted to run a neutralization study. And this other resource I wanted to highlight is CollabFold. So I am by no means a computer scientist. I have no idea how to do, protein folding or any of this, AI stuff. But CollabFold is this really easy Google Colab notebook that you can put in an amino acid sequence and run AlphaFold basically through your browser. I've made fusion proteins this way and just sort of seen, do I attach to the C terminal or the N terminal? How does it look like better one way or the other? So this is a great resource. For those of you who are maybe trying to design RNA or DNA-based vaccines, there's this website, mRNAID, which was developed by Merck. It's an open source tool for optimizing nucleic acid sequences to sort of boost their immunogenicity as well as their expression. And then, of course, the Koch Institute. We have a ton of great cores here with knowledgeable staff, as well as tons of faculty right in the building that know a lot about immunology. So there's tons of great resources here. And so with that, just a few discussion questions to maybe get anything started. If people have any other things they want to discuss about maybe just where they sort of see the next generation of vaccines going and any ideas that people might have.